All right, welcome to week 3 PS Pain Relief slides. We'll be talking about genetics, genomics and medicine and metabolism. I'm Victoria and I'll start off by talking a little bit about genomics and its relevance to medicine. So first of all, let's just talk a little bit about monogenic disorders. So monogenic disorders are any disorders that result from a single defective gene, and they're usually rare. So they can be linked to either the dominant gene, the recessive gene, they can be X-linked or Y-linked or mitochondrial. And these are what we call inheritance patterns. And you can identify these inheritance patterns by looking at pedigrees. And you would have done some pedigree work in Year 12 biology. So the Human Genome Project, just a little background information. So since humans are 99.9% identical, then it's pretty reasonable to have like a template sequence of the full human genome sequence. So that's what the Human Genome Project did. They basically mapped all 3 billion base pairs of the human genome. And when you have this template, it helps you identify the genes that cause disease and lead to susceptibility. All right, so moving on to SNPs, or single nucleotide polymorphisms. SNPs are when a single nucleotide, so your A, T, C, G, differ between people. So recall that the human genome has about 3 billion bases, and for every 1,000 bases, you'll have a 1 base difference. So that is the 0.1% variation between humans. Now, for a variation to be considered a SNP, it must also occur in at least 1% of the population. So if it occurs in less than 1%, then it's not significant enough to be considered a SNP. And SNPs can occur in exons, introns, or between gene sequences. Okay, so these are just some SNP statistics. So around 50% of SNPs are in the non-coding regions. And if these occur in the non-coding regions, and they can impact promoter activity or pre-mRNA conformation or stability. Around 25% of SNPs cause missense mutations. So that will actually mutate the protein and impact its ligand binding, subcellular localization or stability. And then the remaining 25% of SNPs are silent. So you don't see a problem with gene expression in that form of when a SNP is silent. So the genome-wide association studies, these studies helped to correlate SNPs with common diseases using large sample sizes. So they got thousands of cases, so thousands of people with a certain disease, and then thousands of controls. And they basically just mapped out their DNA. In this little graph here, I'll try and draw. So in this little graph down here, you can see that there's like certain dots that align to be higher than others. So this basically indicated that there were SNPs here that were more frequent in the people with the disease. So then that led scientists to believe that these SNPs were responsible in either causing the gene or causing the disease, or causing someone to be susceptible to the disease. Right, so I'll just get rid of that. Okay, let's move on. So SNPs are important in pharmacogenetics. Pharmacogenetics is basically the prediction of drug responses. And it's thought to be 20 to 95% variability in drug responses is due to your genetic code. So for example, warfarin. Warfarin is an anticoagulant that's used to treat thrombosis or blood clots or prevent stroke. Now, different patients have different responses to warfarin. Too high of a dose makes the patient become at risk for bleeding, and too low of a dose, well then it will be ineffective. So what scientists discovered was that the genes CYP2C9 and VKO-RC1, these genes were largely involved in warfarin metabolism and activity. So people who have these two variations of the gene will require a lower dose of warfarin. And genetic tests also exist for these genes to estimate the correct dose of warfarin. So you just need to be aware that SNPs are important in medicine because it helps you determine the dosage for a patient. So another example of how SNPs are important in pharmacogenetics is when you look at your beta-2 adrenergic receptors. So these receptors are present in your sympathetic nervous system, which are your fight or flight responses. So in terms of what the sympathetic nervous system does to your body, when it's activated, your airways will open up. And this is important in people who have asthma, okay? So if you have asthma, you want to open up those airways, so you want to stimulate your sympathetic nervous system, okay? So in other words, you want to stimulate your beta-2 adrenergic receptors, okay? And the drugs that do this are the agonists. Now, SNPs in this receptor will impact the efficacy of these drugs. So albuterol, for example, which is used to treat asthma, albuterol will stimulate your sympathetic nervous system by stimulating your beta-2 adrenergic receptors, okay? And because it stimulates them, therefore you call it an agonist. So SNPs in this receptor will impact the expression of the receptor, and therefore it will impact how albuterol interacts with this receptor. And that's it for this section, really.
Okay, so population genetics. I am not sure if you guys have had this lecture yet, or if you guys are about to have it this week, because as you know, the timetable has sort of been thrown out of whack. And I understand that it's a bit, it's hard, yeah. But hopefully this will give you guys an overall idea of what is in population genetics. So you guys could revise, or you guys could read ahead and just, be a bit more prepared for when the rest of the content is taught over the week. All right, so population genetics, what do we have? We have terminology, we have the Hardy-Weinberg equilibrium, deviations from the Hardy-Weinberg equilibrium, and that's about it. So the basics, yes. So the basics are just these definitions here. So genotype frequency, phenotype frequency, and allele frequency. What are they? So phenotype means an observable physical trait. A genotype is a combination of alleles, and this combination of alleles is what produces the phenotype. And the allele frequency, so alleles are basically alternative forms of the same gene. If you think about eye color in humans, so there is a gene that codes for the eye color in humans, and so the alleles of that gene maybe for brown eyes and for blue eyes. Yeah, and so when you look at the definitions over here, frequency basically means how often you see that specific trait, that specific combination of alleles, or that specific allele itself. Okay, and the Hardy-Weinberg equilibrium. So what is the Hardy-Weinberg equilibrium? So basically, if you remember a bit of probability from year 12, p squared plus 2pq plus q squared equals 1. So 1 is basically the one is sort of the 100% probability. If you look at it here, p squared, 2pq, and q squared, these are probability values. So p squared is homozygous dominant, so that's p times p. So dominant times dominant. So the chance that you'll have a dominant times the chance that you have another dominant. Again, 2pq, heterozygous, so pq plus qp. So the reason that this is 2 rather than just P times Q is because you have a maternal gamete and a paternal gamete, right? Sperm and egg must create new person. So you can see it as 2PQ being PQ. So maybe this is maternal P times paternal Q plus maternal Q times paternal P. That was a mouthful. Okay, so if you see it that way, as it being split between maternal and paternal and the different combinations that you can have, you'll kind of get why this is 2 rather than just PQ. Okay, so 2PQ is therefore the probability that you will have a genotype that's heterozygous, and Q square. So again, that's the chance that you have a recessive allele times the chance that you have another recessive allele. So this gives you the chance that you'll have a homozygous recessive genotype, and all of this adds up to 1 because 1 is 100%, and you have a 100% chance of having a genotype. If you are a living thing, congratulations, you have a genotype. All right, moving on. Deviations, right? So not all populations follow the Hardy-Weinberg equilibrium. As with everything that's mapped out on paper, it works on paper, but not often in real life. So this may signify new genetic variations. So this is caused by mutations or changes to existing variations. So this can be done by having gene flow, positive or negative selection, genetic drift, and non-random mating. So the following slides will just be a very brief overview. They will not go into a lot of detail because it is important that you guys understand the concepts. And I have an email. It is important that you guys understand the concepts and the details. I trust that you guys have whatever it takes to fill in the details yourselves. This is just succinct, revision focused. All right, okay, so how can we deviate from the Hardy-Weinberg equilibrium? So mutations. So there are 2 types of mutations: point mutations, which is basically when there's a change in a singular base or a singular nucleotide, or chromosomal mutations. So this is when there are changes in segments of a specific chromosome. All right. So frame shift mutations. What is a frame shift mutations? It happens when the reading frame of a codon is shifted. So this can happen when you have an insertion. So you add another base right in front of a codon, and therefore, so this is your codon. If you add another base, this becomes the first one, 1, 2, 3, instead of our original 1, 2, 3, right? So now this is the first codon, and this becomes part of a second codon. All right, I just look like I'm throwing up gang signs. Okay, so that's kind of what it means by a frame shift mutation. And as you can see, as you may have been able to see, when you have a frame shift mutation, this shifts the reading frame for every single codon following that point where there was an insertion or a deletion. So if you can already guess that this means that large portions of the sequence of genes is going to be affected. It's going to be completely changed. You have completely different amino acids being synthesized. You have a completely different product being formed, and you have a completely different functionality of that product. And oftentimes this means that this product is non-functional and it just can't do anything. All right. Silent mutations. On the other hand, if you have your three bases in a codon, the first one, second one, important. The third one, not so much, right? Because if you remember, don't know if you guys have been given a table of all the codons, all the amino acids in our human body. Every single amino acid is coded for not by one codon, but by a set of a few codons, where the first and second letters are the same, final letter is different, right? So this just goes to show that changing the third base of a codon changing the third base of a codon is not very consequential to the cell at all. Silent mutations can also happen in non-coding DNA. So if you just randomly switch out a nucleotide in a section of DNA that's non-coding, who cares? It doesn't code for anything. And so whatever change that's happened in, a non-coding region. Nothing happens. So this just leads to polymorphisms. Earlier in the section, you've been told about single nucleotide polymorphisms. These do not have any effect on our functioning as human beings, as living organisms. However, they just lead to a bit of genetic variation, but not phenotypic variation. So next up, missense. So missense. As I've said earlier, the first and second bases of a codon are very important. And when you change these two, you essentially change the amino acid that's being coded for. So when you change the amino acid that's being coded for, once again, you have a different amino acid sequence, different protein that has been formed, different product, different functionality, and you can either have a protein that is still kind of functional but just has a different structure, or you can have a protein that is essentially non-functional entirely. So not a good time, basically. And then lastly, nonsense mutations. Nonsense mutations, they are no laughing matter. So what happens when you have a nonsense mutation? You have an inappropriate formation of a stop codon. So if you remember your stop codons, they are UAA, UGA, and UAG, right? Okay. And if you were to happen to switch out a random nucleotide to one of these, you accidentally happen to form one of these stop codons. This would, sorry. This would mean, that you would form a stop codon, and the translation process would terminate right there. So you would have premature termination of the translation process, and this would lead to a protein with a loss of function. So obviously that's not good also. Chromosomal mutations, very quickly, there are deletions. Where you lose segments of DNA, there are duplications, there are inversions, and there are translocations. Well, it's pretty straightforward. So you can lose a segment of DNA. A segment of DNA can accidentally be replicated twice. You can have a segment of DNA that breaks off, kind of flips around, and then rejoins in an inverted form, and that can lead to this can also lead to loss of function. And translocations happen when DNA segments are joined to another chromosome. So let's say you have two chromosomes. This section breaks off, and then they just kind of switch around, and they form two new chromosomes with a segment that doesn't belong. So you can see these in Philadelphia chromosomes. Look those up, homework for tonight. And these can lead to deletions, duplications, and sterility. So again, not a good time. But the bright side is that these mutations will rarely actually affect populations dramatically. So even though some of these mutations will lead to loss of function or sterility, they will rarely affect the populations that badly. All right. So yeah. So next up, how else can we deviate from the Hardy-Weinberg equilibrium? Well, we can have gene flow. So that's caused by a migration of individuals between different populations. And when individuals from different populations migrate freely from here and there, this means that they carry their gametes with them as they move. If they reproduce with individuals in a new population, this means that their new set of alleles that they have carried from like a foreign population are being passed on to the next generation. So the next generation has increased genetic variation because they now have some new alleles being introduced to them by the foreign individuals. Yep, yep. So positive and negative selection. So some phenotypes naturally confer higher or lower levels of reproductive fitness. So just think of the type of person that you want to get jiggy with, right? You obviously have some specific traits. You know, you have specific traits that you say, oh, deal breaker, or no, hell no. And when you think like that, you are actually conferring reproductive fitness onto these specific traits. Think about it. Oh, that's so cool. All right, so higher or lower levels of reproductive fitness will lead to a higher or lower chance of these individuals with those specific traits mating and passing down their alleles to the next generation. So for example, you have birds that have brightly colored feathers because they want to attract a mate, but then some birds don't have that bright of a color of their tail feathers, and so they don't have as much, as much game, right? So they don't pass down their alleles, and so um, and so bright colored tail feathers are overrepresented in the population because the females only like, the dudes with the bright tail feathers. There's also heterozygous advantage in people with sickle cell anemia. So sickle cell anemia, not a good time, but the bright side of that is that you can have immunity to malaria. So the reason why, so even though sickle cell anemia is not a good time, it still remains strong in many different populations because these individuals are immune to malaria and therefore have some form of higher reproductive fitness in them, which is, good. You go, people with anemia. And then fourth, genetic drift. So this is caused by a sudden reduction in the population. And bear in mind that this sudden reduction in the population is random. So this leads to the new population not being representative of the old population. So the old population may be representative of the Hardy-Weinberg equilibrium, but the new population may not, because it's just been randomly sliced at random. So one example of this is the founder effect. So some individuals may migrate to a new location, and this founding group, such as the cast of Hamilton, is randomly assembled and therefore not representative of the original population. So group of individuals may just randomly decide, I do not enjoy this environment, and they migrate somewhere else. And this group of people this group of individuals that leave the original population, there is not much intentionality in who they are. So they're a very random group, not representative of the original population. So when they multiply back into a larger size, they do not follow the Hardy-Weinberg equilibrium. All right, so next, genetic drift. Again, another example is a population bottleneck. So this is when a catastrophic event wipes out part of the population at random. Sounds familiar? Ayo, my man's wiping out 50% of the population at random. So when these small populations grow into full-size populations, the original genetic variation is no longer representative of the original population. So if half of the world has been snapped out, and then 5 years later, the people that have survived the snap kind of mate into a slightly larger population. This slightly larger population is no longer representative of the original population that was, happening before the snap. Okay. And last but not least, non-random mating. So inbreeding usually leads to homozygosity. Outbreeding usually leads to heterozygosity. And yeah, roast me for the image choice. I invite you. Okay, well, on a more serious note, if you're a dog person, and you like Labradoodles, Labradoodles are the result of outbreeding because we have Labradors, we have Poodles, we put them together and we have a new phenotype, a new genotype, in fact. And so this leads to heterozygosity. Inbreeding, you think of those dogs at those fancy British dog shows where you want a pure blood border collie or something like that. And so that would mean that this pure blood border collie would only have to have mated with other pure blood border collies and have the most homozygous of homozygous genes because it has only been inbred and inbred and inbred and inbred. So that's just the picture you want to have in your mind. Honestly, a lot of this is very commonsensical. As long as you understand the general concept and can apply it, then you should do just fine. So that's it for population genetics. Thank you very much, guys.
Guys, I'm Vicky, and I'm going to take you through a few of the metabolism topics. This is my first time recording or even doing a PSP in general, so bear with me. So we're going to start with carbohydrates. So what are carbs? Carbohydrates are basically molecules that are made of carbon, hydrogen, and oxygen, and the H2O ratio is always 2:1. And this table shows you a few of the carbs. And if you've done year 12 bio, this should be revision. So monosaccharides, the basic building blocks of carbs. Disaccharide is 2 monosaccharides, and polysaccharides are more than 2 of them. So glucose is a 6-carbon sugar. It is the form that the body absorbs, and it can be transported in the blood. Disaccharides, not so important. And polysaccharides is starch, glycogen, and cellulose. So yeah, in year 12, you'll learn things like, which ones are branch, which ones are linear, which ones are whatever. But what's important is to know that glycogen is a form of carbohydrate that's stored in the body. And that cellulose is basically like dietary fiber that you like poop out. They're the ones that the body can't digest. Okay, so this is pretty important as well. So the source of carbohydrates in the body is either by your diet, so the breakdown of carbs and starch, or it is a mobilization of what is already stored in your liver and your muscles. And this diagram here is pretty important to remember. So throughout the day, of course, there's the dietary one, which you eat. So it comes, that's why you'll see in this diagram, it corresponds to pretty much like what time you eat. So this would be breakfast, this would be lunch, that'll be dinner. And so between the meals, it is this dark gray curve. So this is glycogenolysis. So it's a breakdown of the store in your body, the glycogen. And then what you need to know is that after 8 hours, this number is pretty important. So after 8 hours, that's of like not eating, that's when you use gluconeogenesis. So for example, after your last meal, you use the glycogen here. And then this is around this time when you're sleeping, where your body actually makes, like synthesizes glucose to keep, as like an energy source. Yep, okay. Okay, so this is also revision from year 12 bio and not that important. So first of all, your body will like digest when you're eating, your body will digest carbs and until they're only monosaccharides because that's the only form that can be absorbed from the lumen of the gut into your blood. And therefore there are various enzymes in your body to break down different forms of carbs. You don't need to know exactly which kind, that's why I've only given a few examples here. So for example, maltase for maltose, lactase for lactose, yeah, stuff like that. And then in your gut, there's also these brush border enzymes that's in the microvilli of your villi in the intestine that helps you digest these things. And then for the absorption, these are basically, well, Nirma didn't really teach it. I don't know if you guys still have Nirma this year, but these are this content was basically a bunch of videos on Moodle that they wanted you to watch. So it's pretty low yield, but you'll see that different molecules absorb like different ways. They're absorbed different ways into your body. I'm not going to really go over them because you basically won't be tested on that. And it's, yeah. Okay, so this is more important now. So this is the mobilization of stored glycogen in your body, also known as glycogenolysis. So this is signaled by glucagon and adrenaline when blood glucose is low. So glucagon is basically something that's synthesized in your pancreas. When it realizes that your blood glucose is low and that you need energy. So first, glycogen is converted to gluon phosphate by this enzyme called glycogen phosphorylase. And then it becomes from gluon phosphate to glu-6-phosphate, so G6P, by another enzyme, which you don't really need to know. And then this part's important. So it depends on which organ this is happening in. So if this is in your liver, this G6P is going to be further broken down into glucose by this enzyme called glucose 6-phosphatase. This enzyme is only in the liver and in the kidneys. And then since G6P is converted to free glucose, it can be transported into anywhere in the body through your bloodstream. So, for example, to supply the brain. On the other hand, in the muscle, the G6P that's converted from glycogen is used right away. It directly enters glycolysis and is used as an energy source in your muscle. This is because there is no enzyme, glucose 6-phosphate, in your muscles. And so therefore this glucose is used right away and can't be exported to anywhere else in your body. Okay. Now is the breakdown of carbs. So cellular respiration. It's pretty complicated. There are a few steps. So there's glycolysis, and then there's fermentation in anaerobic respiration. And if it's aerobic respiration, you'll first go through the link reaction or pyruvate decarboxylation into the citric acid cycle and then to the electron transport chain. And on this side, I've got where these various steps occur in the cell. So the glycolysis occur in the cytoplasm. Pyruvate decarboxylation occurs in the mitochondrial matrix, CAC in the matrix as well, and lastly, the ETC in the mitochondrial inner membrane. So I'm only gonna go through, I think, the first three with you. These three with you, because I'm pretty sure Mayanne is have already gone through last week, the CAC and the ETC, so. Okay, so the first thing is glycolysis. So it's the conversion of glucose to pyruvate. So glucose is a 6-carbon molecule, whilst pyruvate is a 3-carbon molecule. So unlike biochem students, we don't need to know the individual steps. They're just a few things to memorize. It's an exergonic reaction. It can occur whatever the condition is, whether with oxygen or without oxygen. And there's a small yield of energy in the form of 2-ATP. So basically, it's a 10-step enzyme catalyzed reaction. So the first 5 stages is called the preparatory phase, and then the last 5 steps is called the payoff phase. Okay, and steps 1 and 3 consume ATP, while step 4 breaks glucose from 2 to 3. Breaks glucose into 2 times 3 carbon molecules. So 2 3-carbon molecules. And step 6 releases 1 NADH, and step 7 and 10 releases 2 ATP each. To be honest, we weren't tested on the individual steps, but I think these things are good to know and just memorize. But you don't need to memorize in detail as the diagram on the right. And so you'll see that the input is 1 glucose here, and the input is 2 ATP as well. So in these steps, you need 2 ATPs to basically break this molecule down. And the output is 2 pyruvates. So after this 6-carbon molecule is broken down to 2 3-carbon molecules on each side, there's 2 pyruvates, so basically it's another pyruvate on this side. Like this side happens twice. And then, so after in the second 5 steps, there is a yield of 2 ATP, and since it happens once again twice, with each 3-carbon molecule, and in total there's a 4 ATP. And that's why, but since like 2 is already used at the start, that's why your yield is just 2 ATP in total. And there's also 2 NADH that will be produced in the process here, the one on this side, one on the other side again. And these are coenzymes that carries electrons that will later be used in the electron transport chain. All right, so now we're going to move on to the phase of pyruvate, and it's different depending on the availability of oxygen in the body as well as NAD+. I'm going to start with anaerobic, so that's where there's like a lack of oxygen in the body. So in humans, pyruvate is converted to lactate using the enzyme lactate dehydrogenase. And this reaction doesn't produce ATP, but it kind of contributes to energy in its own way because it produces NAD+, as you can see here, which is used in glycolysis. So it kind of contributes to energy that way. So lactic acid, the same as lactate, well, it's acidic, so it lowers blood pH, causing lactic acidosis. So this process occurs vigorously in contracting muscles and also in red blood cells. You'll later learn why it happens in red blood cells and some other cells. So this is kind of like when you're running, your muscles get sore. It's just kind of like the buildup of lactic acid. And in aerobic respiration, so where there's oxygen, pyruvate enters a link reaction. The link reaction is also known as pyruvate oxidation or pyruvate decarboxylation. So first, the pyruvate that was in the cytoplasm of the cell diffuses into the mitochondria, and then it's converted from pyruvate to acetyl-CoA. So once again, it's a series of steps. First of all, this is catalyzed by pyruvate dehydrogenase, which is an enzyme that uses thiamine. So this is a kind of vitamin B. And pyruvate is combined with coenzyme A. And this process also loses 1 carbon. And this process is called decarboxylation. So because it loses 1 carbon, you know how pyruvate is a 3-carbon molecule? It then becomes a 2-carbon molecule. And this step also produces another NADH. So this whole link reaction occurs once with 1 pyruvate. So basically it occurs twice with 1 molecule of glucose, since 1 molecule of glucose produces 2 molecules of pyruvate. So yeah. Oops. Okay. Next, we're gonna move on to the synthesis of carbohydrate. So it's gluconeogenesis. As you may remember, this occurs in the body after 8 hours of not eating. This process only occurs in the liver, and it is used to maintain blood glucose levels during the fasted state when hepatic glycogen is depleted and no free glucose is available anymore. So this is stimulated by glucagon produced by the pancreas, and it's basically the reverse process of glycolysis. You really don't need to know this in much detail at all. Which is, that basically it's kind of like the opposite of glycolysis, but there's a few differences. For example, the 3 steps, which uses different enzymes and differs from glycolysis. And any step after pyruvate can't be reversed. And oxaloacetate is a product of one of the subs. So this synthesis, the synthesis of carbs can be done by pathways using different starting materials. This includes amino acid, lactate, and glycerol backbone that is originated from triglycerides. And glycogen occurs when blood glucose levels are high after amino, and insulin produced by pancreas signals from muscle and liver cells to uptake blood glucose and store it as glycogen using glycogen synthase.
Hello Jaffies, my name is Christian and I'm going to be talking to you guys about lipids. So here we go. Now we're going to be learning about how do we synthesize lipids. We're going to be learning about the structure and the breakdown of lipids in the body, and we're also going to be learning a little bit about these things called lipoproteins right here. So let's get right on into it. My goal is to help you guys to understand this U-Butte concept map put together by our very own Mayanne. Now, all that this concept map is really telling you is it's telling you, okay, around here, the body synthesizes its own lipids and then sends them via this pathway into storage here, okay, in our fat cells. And the other way that we get fat there is by eating it, and that's what this pathway is talking about. We'll get to each of these steps in due course. And the final little bit down here is talking about once it's in storage, how do we then use it? So that's just right here. Now let's get right on into how this all works. There's three types of lipids that concern you guys. Lipids are just biomolecules that are soluble in non-polar solvents, or basically not water. So they're not soluble in blood, for instance. Now, the three types that you guys need to know are triacylglycerides, so TAGs, we often abbreviate this to. These guys are made of 3 fatty acid chains connected to a glycerol. Then we have our phospholipids as well, which instead of being 3 fatty acid chains, we have 2 fatty acid chains, plus a glycerol and a phosphate. So you might remember phospholipids make up that phospholipid bilayer, and they've got a polar region and a non-polar region. And then finally, we've got sterols. These guys are a little bit different. They've got lots of hexagons and things like that in their chemical structure, and their synthesis pathway is very different to these two. But one thing that these guys all have in common is that they can all be synthesized from this base molecule here, acetyl-CoA. You might remember this guy from the Krebs cycle. Now, the real important part of this lecture is that you understand how central acetyl-CoA is to our body. Okay, that's the most important thing that you can take from it. This is like the biochemistry 101 right here. Okay, so we're going to start by looking at this little section of the diagram first. What this is talking about is how do we use acetyl-CoA to produce triacylglycerides, that first kind. Now, I should mention TAGs, those ones are used to store energy, okay? So let's have a look at how we get TAGs. So there's 2 parts. We need our glycerol and we need our fatty acids. Now, the glycerol is synthesized by the liver in pathways that I've listed here, but I won't talk about them in any detail. But in terms of our fatty acids, that we have to know. They're synthesized from acetyl-CoA, as we've discussed, but we also have to eat some, and those are essential fatty acids. I'm skipping those slides today. Let's talk about how the body synthesizes its fatty acids. Now, what it involves is this intermediate step here, okay? This little red boy here, malonyl-CoA, is a 3-carbon molecule. Acetyl-CoA is a 2-carbon molecule. So first there's a conversion step to get that 3-carbon molecule, and then that 3-carbon molecule is joined with other molecules of its kind. So lots and lots of malonyl-CoA is joined together, and we get fatty acids. The tricky thing is that instead of getting 3 carbons at a time, we actually only get 2 from the malonyl-CoA, and then another carbon goes into other stuff in the cell. We don't need to know it in any more detail than that. Now, you guys might have heard of beta oxidation. This is not the reverse of beta oxidation because it involves different enzymes and it's a completely different pathway. But it does effectively do the reverse of beta oxidation. Now, a common pattern you'll find in biochemistry is that when we have anabolic reactions like this, reactions that are building things that are using energy, we can only do them when we have a diet that is rich in energy, because then there's lots of energy available to use for this storage, okay? So if we have a diet rich in energy, then we're going to produce more fatty acids and more fats. On the flip side of that, if we have a diet that has less energy, we're not going to produce as much fatty acids because our body is using that acetyl-CoA in the Krebs cycle rather than synthesizing more fatty acids. All right, and I'm just saying here that it's worth knowing malonyl-CoA because Nirma seems to think it's very important. It's in her slides. And the final step of triacylglyceride synthesis, so once we've synthesized the fatty acids via malonyl-CoA and then that big pathway that we discussed before, and once we've got our glycerol from the liver, the only remaining step is to join those two together, and the only thing you need to know about that is that enzymes do it. That's the only level of detail you need. All right, now we're going to talk about how the body synthesizes cholesterol. This is a completely different pathway to fatty acid synthesis. However, it does still involve acetyl-CoA, as we can see here. The only thing you need to remember about this is that there's a very crucial enzyme here, HMG-CoA reductase. And this enzyme is important because it's a target for certain drugs involved in cholesterol management, which you'll learn about a little bit later in the year. So HMG-CoA There, HMG-CoA reductase allows acetyl-CoA to be converted into cholesterol. So when it's active, more acetyl-CoA gets converted into cholesterol. Now, similarly, because this is an anabolic reaction that requires lots of energy, it's only going to happen when we have a diet that's rich in energy. Now, in here, you'll see our markers for that are insulin and glucagon, but it makes sense because if you have elevated levels of glucagon, that means you've just eaten. So you have lots of energy available because you've just eaten to make more cholesterol. Conversely, if you have high levels of glucagon, well, you release glucagon in between meals. So you're in a fasting period. So your body says, hang on, I'm going to slow down the cholesterol synthesis now, because if I keep producing it, I'm going to deplete all my glycogen stores. Because remember, glucagon is breaking down your glycogen. And then if you are synthesizing cholesterol, you're going to deplete your glycogen stores. So that's how this regulation pathway works. The most important thing you can know is none of this business down here. This is completely unimportant. Just know that insulin increases the rate of cholesterol synthesis and glucagon decreases it, and it's regulated by this enzyme HMG-CoA reductase. All right. So we've spoken about how we get cholesterol and how we get fatty acids from acetyl-CoA. Now I'm going to talk about how do we get fats that we eat into our fat storage. And we'll talk a little bit later about how we get this fatty acid, how we get this fat and this cholesterol into that same place. But for now, let's talk about the ones that we eat. Okay. So first we have to digest and then absorb the fats that we eat, okay? And to do that, we have these enzymes lipases. So ASE is that enzyme ending. It means it breaks down fat. And we have a few important ones in various parts of the digestive tract, but the most important one is in the pancreas. It has these bile salts and this pancreatic lipase that breaks down the triacylglycerides into fatty acids and glycerols, okay? And so we can see we have our fatty acids floating around free of their glycerols just here. Now the micelles, not massively important, but worth knowing that they exist and that they're an intermediate step in this, okay? Now, if you remember that fatty acids are very nonpolar, then we're going to remember that they diffuse really easily across our cell membranes. And in this case, it happens in our intestine, in the intestinal epithelial cells, which we call our enterocytes, okay? So they diffuse really easily across into our enterocytes. That's the fatty acids that do that. In there, they get converted back into triacylglycerides, and they get packaged up into these little chylomicrons. Pretty funky word, and I remember they confused a lot of people last year, so I'm going to try and explain them in a bit more detail. All they are is this big ball of fat, right? So they've got phospholipids on the outside, as you can see, a single layer, not a bilayer. And then inside, we've got a whole bunch of triacylglycerides, as you can see. And finally, it's studded with these apolipoproteins, shown in blue here, okay? So we've got phospholipids studded with apolipoproteins with a triacylglyceride core, predominantly. Now, the whole thing together, we call it a lipoprotein, and there's a few different kinds of lipoproteins, and chylomicrons are just one kind. So we'll talk about that in a moment. Now let's talk about what these guys do. What they're doing is they're taking the tags, the triacylglycerides that you eat, and they're packaging them up into a nice ball and delivering them to the tissues that need tags, the tissues outside of your liver. So we call them extrahepatic tissues. So when they do that, they actually react with something called lipoprotein lipase. Not a massively important step. It's in a diagram a bit later, you'll see. And when they do that, they convert to these things called chylomicron remnants, which go to the liver for further processing, okay? So in other words, most of the fat that you eat will go to your cells, and whatever's left over goes to the liver, okay? And so that's what's happening here. That's what's happening in this part of the diagram. We eat fat, it ends up in our chylomicrons, which goes in the bloodstream, released by our enterocytes, goes into the bloodstream, into storage. It may release a bit of cholesterol into our body cells, and finally, whatever's left over in chylomicron remnants goes back to the liver. Now we're going to learn about how do the tags that we make and the cholesterol that we make in our body, how do they end up in storage? So what's happening, right, is we have these VLDLs which are produced by the liver. Now you might be wondering, why do we need VLDLs? Okay, perhaps I'll explain a bit more what they do first. they transport tags, triacylglycerides, to tissues, but that's exactly the same as what chylomicrons do. They transport tags to tissues, right? Well, the difference is that these guys come from the liver and they're released in between meals. So chylomicrons you get as you eat, because the fat that you eat ends up in your enterocytes, gets absorbed, and then gets packaged into chylomicrons, whether you like it or not. But your liver can regulate the amount of fat that you have in your bloodstream and available for your tissues via these VLDLs. So that's what it's doing. It's making sure that you have enough fatty acids for your tissues, even in between meals. So very important job. Now, that's what it does. It delivers tags to the tissues. And when it does that, whatever's left over can either go back to the liver, or it can get converted to these low density lipoproteins, LDLs, okay? Now, VLDLs are mostly tags, but LDLs are mostly cholesterol. So cholesterol is very important for your tissues as well. But you guys might have heard about bad cholesterol and things like that. Well, that's essentially LDL. If you have high levels of LDL, it's very bad for you. You can get this thing called atherosclerosis. But don't fall into the trap of thinking that that means cholesterol is a bad thing. It's still very, very important, as we've discussed. Now, the final one that we have, it's not included in the big concept map because it would make it a little bit too arrowy, but we have HDL, high density lipoproteins. These guys, they basically are like little cleaner molecules. They go around and they pick up little floaty bits like this of LDL, not of LDL, of cholesterol, and they take it back to the liver. And that's very important because if you have too much cholesterol building up, you can get atherosclerotic plaques in your blood vessels. So HDL is basically reducing the risk of that. If you have high HDL, it's very good. If you have high LDL and low HDL, you may get atherosclerosis. So here's an easier version of Nermer's scary diagram. I'll go through it really quickly. Basically, what's happening is you eat, the fats that you eat get packaged into chylomicrons by your enterocytes. Those chylomicrons get released into your bloodstream. They deliver fat to your cells via a reaction with lipoprotein lipase, not massively important in the intermediate step, but the role of chylomicrons is important. They deliver fats to cells, and you get chylomicron remnants, not labeled, but it's this pink arrow here, which goes back to the liver. Those chylomicron remnants and other fats that the liver synthesizes get packed into VLDLs, which contain basically triacylglycerides. And they follow much the same path as chylomicrons because they're doing the same job. They deliver their tags to cells, and what is left goes up here. It can either return to the liver, or it can be turned into LDLs, which are mostly the cholesterol that's left over in the VLDLs. All right, so they get converted to LDLs and they deliver cholesterol to cells, not massively important, but it's via receptor-mediated endocytosis. And whatever's left goes back to the liver for further processing. So that's kind of the way the cycle works. And then finally, we have HDL, just clearing excess cholesterol. Okay, finally, we're going to talk about this final bit, mobilization of tags. It's pretty straightforward. There's 5 central steps. First, I haven't included this step, but it's hormone regulated. A hormone comes along and it tells your fat cells, your adipocytes, to start breaking down your triacylglycerides. And it does this by activating this tag lipase here. So the triacylglycerol lipase, or you can also see it here on the diagram, breaks down the tags into fatty acids, which you can see here, and glycerol here. Now those fatty acids, as you know, they're very non-polar. They diffuse across the adipocyte membrane very easily. But if they just went straight into your blood, well, blood's aqueous, they would just clump together because they're non-polar. So the non-polars like each other. To prevent that from happening, we have these proteins floating around in our blood called serum albumin, okay? And the fatty acids bind to the serum albumin so that they don't clump together too much, basically. The serum albumin is like a little ferry boat for these fatty acids. And then the serum albumin then delivers the fatty acids to the target tissues via the blood, where they dissociate from the serum albumin, and the fatty acids then just enter the cells into the cytosol to be used. So that's all there is to know. Just the only really important thing to know is the crucial role of albumin in this equation. It binds to the fatty acids and is a little ferry boat for them. All right, so here's the whole diagram, okay? What we've learnt today is that you can synthesize cholesterol and triacylglycerides from acetyl-CoA. There's a few steps involved. In here, HMG-CoA reductase, and here malonyl-CoA is the intermediate step. From there, we have these lipoproteins, particularly VLDLs, that send these bad boys via the blood to the tissues. And we can also get cholesterol delivered to the tissues as well. The other way that we get fat into our cells is by eating them. They turn into chylomicrons. The chylomicrons deliver fat directly to tissues, but they also turn into chylomicron remnants, which go into the liver and get processed via the endogenous pathway here. And then finally, when we need to use them, they break down due to triacylglycerol lipase, and then those fatty acids bind to serum albumin and go to the target tissues. Now, when we break down our fats, you guys may have already learnt this. Basically, all that's happening is that we're turning these long fatty acid chains into acetyl-CoA. Now, acetyl-CoA is a 2C molecule, 2 carbons, so we take off 2 carbons at a time, as you can see. So beta oxidation is the repetition of the removal of 2 carbons from this fatty acid chain over and over until you're basically left with only 2C units. And every single time that you do a chop and you take off 2 carbons, you get an acetyl-CoA, an FADH2, an NADH, and an H+. But the thing is, on the last pass, you're chopping a 4C molecule in half to get 2 units of 2C. And as a result, you can make 2 acetyl-CoAs out of it. So on the last pass, you get an extra acetyl-CoA, but you only did 1 chop, so you still get 1 FADH2, 1 NADH, and 1 H+. So to extrapolate that, if you had an 18 carbon fatty acid and you broke that down via beta oxidation, you would get 8 FADH2, 8 NADH, 8 H+, but 1 extra acetyl-CoA, 9 acetyl-CoA. Now, acetyl-CoA from beta oxidation just goes into the Krebs cycle or the citric acid cycle, as you can see here. And all these other things, this FADH2, the NADH, and the H+, go into the electron transport chain here, either as loaded electron carriers or to react with oxygen here to make water. That part is not hugely important. It's more important to know that acetyl-CoA goes into the Krebs cycle and produces a whole bunch of loaded electron carriers. All right, that's it, everybody. I hope you enjoyed.
All right, okay, so proteins. Now, proteins are what you need to get positively massive. You all know that just fine. So, proteins, ironically, is not that massive of a topic. I do feel like lipids would have been actually the most important one. Aside from carbohydrates, proteins are not that big of a topic. But still, you should still learn it because everything here can be tested. So without further ado, digestion and absorption. So here I have a very helpful diagram showing you how proteins are digested and absorbed. Basically, in the stomach, you have proteins that are broken down into short polypeptides. So you remember that in your stomach, you have hydrochloric acid, you have a very, very low pH, and this helps to really break down your proteins. It also activates an enzyme called pepsin. Pepsin breaks your protein into shorter polypeptide chains. And because it's activated by the acid, it's very, very active in your stomach. So once these short polypeptides get down into your small intestines, the pancreas secretes peptidases or proteases, and these polypeptides are broken down into amino acids. These amino acids, right, they're taken up by enterocytes, which are absorptive cells in the small intestine, and are then absorbed into the bloodstream and are circulated to cells that require them. All right. And how are they utilized? So here, cells receive amino acids from the bloodstream. We know that from the previous slide because I just said it. So what happens to these amino acids at the cells that have taken them up? So they can be transaminated. So if you see here this red one, this red amino acid. We know it's an amino acid because it's got the NH2 group, right? Whereas this one is a keto acid, right? And it does not have the NH2 group. So by transamination, you see that you see that this one with the R2, the one that previously had the NH2, it no longer has the NH2 because this NH2 has been given over to the one that had the R1. And now this is an amino acid, and this one is no longer an amino acid. So basically what happens is that the NH2 just switches between keto acid and kind of confers upon different keto acids the status of being an amino acid, if you think of it that way. All right, so deaminations. The amine group is split from the carbon skeleton. So this is your amine group, right? And then this is your carbon skeleton. Again, this is a very, very clear diagram to show you what I mean. So this amine group, when it's split from the carbon skeleton, it's converted to ammonia, which is bad, which is also, interestingly enough, the reason that pee smells so bad. And this NH3 is converted to urea. Urea is then excreted as urine. That part we know well. And the carbon skeleton enters the citric acid cycle. It can be, as you can see here, it can be broken down into glucose or acetyl-CoA, or even ketone bodies. Ketone bodies, you'll cover it later, not now. So with protein synthesis, sorry, if I could just, well, I can't. Okay. Well, these free amino acids, okay, sorry. Now it's gone. So some other amino acids, if they're not being transaminated or deaminated, they are used in protein synthesis. So these become the free amino acids that are attached to amino acyl tRNAs and are used to synthesize polypeptides during translation, and are used to help cells function, help cells build things inside cells. Wow, that's really cool. Okay, next up. So that was all of it for protein synthesis. Honestly, it's not a very big topic. So dietary proteins, we eat them, we drink them in our massive big boy protein shakes. They get broken down into polypeptides. Remember that happens in the stomach. That happens in the stomach because of hydrochloric acid and also pepsin. Then after that, these pass into the small intestine where they're broken down by peptidases into amino acids. Then they can enter the liver. They would enter the liver first, yes. So some of them just pass through, circulated intact to muscle cells. And these proteins are used as building blocks, helping to build muscle. As you can see, this is Jake getting positively hench. Amino acids, when they pass into the liver, can also be transaminated. And you know that's just the amino group being switched around between different keto acids. An amino acid may become a keto acid, and a keto acid may become an amino acid. Who knows? It's a wild, wild world out there in the liver. And some amino acids may be deaminated. So that's the split between the C skeleton and the ammonia. Ammonia becomes urea. Urea becomes urine. Urine is happening, here. There we go. Mannequin piss, art reference. The C skeleton, on the other hand, is also taken to the muscle cells and used in the citric acid cycle to produce ATP, which is what is powering Jake through his, what I would say is an intensive cardio set. So that's about it for protein metabolism. As I said, not a lot of content in this one, but still important to know. So that's it. Bye.
All right, now we're gonna move on to alcohol metabolism. I've recorded this like 4 times already. It's driving me nuts, but hopefully I got it right this time. So the metabolism of ethanol occurs in 2 pathways, and it occurs in the stomach lining and the liver. So the main pathway, about 90% of it, occurs in the stomach and liver, and it happens when you only drink low to moderate levels of alcohol. So first, alcohol is converted to acetaldehyde by this enzyme called alcohol dehydrogenase. And this molecule is highly toxic to the brain and liver and is also a known carcinogen. And that's why when it gets into the blood, it's associated with hangover symptoms. And you'll note that this reaction is a reversible reaction, as the arrows point both ways. Next, this acetaldehyde is converted to acetate using aldehyde dehydrogenase. This is an enzyme found in the mitochondria. And this one is an irreversible reaction. So you'll note that if we have a deficiency in this enzyme, acetaldehyde can't really be converted to acetate anymore. And this is associated with the Asian glow, Asian flush, whatever you call it, because there's a buildup of this. And the buildup of this also causes a adrenaline release, leading to flushing, nausea, and vomiting. And lastly, this acetate is converted to acetyl-CoA in the liver and the blood. Yeah. And one thing to note is that NAD+ is used in both of these steps to make NADH. This becomes important later. So the other one is the MOES, and it's only about 10% and only happens when you drink a lot of alcohol, and it occurs in the liver. So it uses this enzyme called CYP2B1, which is found in the SCR, the smooth endoplasmic reticulum. And when it converts ethanol to acetaldehyde, it produces ROS, so reactive oxygen species. It's also a really toxic byproduct and can cause things like liver damage, DNA damage, and organelle problems. And then this acetaldehyde is converted to acetate using either the same enzyme again or using ALDH, which is used in the previous pathway, and most occurs with ALDH. And once again, if we go this way, it produces ROS, and if we go this way, it uses NADH to produce NAD. It uses NAD+ to produce NADH. Yeah, and later it becomes acetyl-CoA again, the acetate. Okay, anyway, and something interesting is that this enzyme, the more you drink, the more upregulated this enzyme becomes, and that's how we develop tolerance to alcohol. Okay, so we'll note that both of these pathways lead to the same thing. So it leads to high levels of mitochondria NADH. So like I said, two of those enzymes uses NAD+ to make NADH. This results in a low NAD+ to NADH ratio and high levels of acetyl-CoA, because all the acetate then becomes acetyl-CoA. And this results in a bunch of things that, what is going on? Results in a bunch of things that's associated with all the bad things about alcohol. For example, it causes dysregulation in glycolysis and also causes lactic acidosis. So both of these are caused by the high NAD+ levels. So high NAD+ favors the reverse reaction of glycolysis, because as you may remember, glycolysis needs NAD+ instead of NADH. So it reverses the reaction, leading to reduced glycolysis and increased G3P, which is just one of the products and one of the steps of glycolysis. High NAD+ also favors the reaction pushing the conversion of pyruvate to lactate by lactate dehydrogenase, because this process, instead of using NAD+, it uses NADH. So yeah, so lactate, this leads to lactic acidosis, where your blood is like too acidic. And it also alters the Krebs cycle, as the NAD+ is needed in the Krebs cycle as well as glycolysis. It also suppresses gluconeogenesis, because pyruvate is something needed in gluconeogenesis. And as we see up here, the pyruvate is being converted to lactate instead. So this leads to hypoglycemia, so low glucose levels, and why you feel tired after like a day of drinking. And lastly, it leads to increased fatty acid synthesis and a decrease in fatty acid metabolism, because there's such high levels of acetyl-CoA that it pushes the reaction converting it to malonyl-CoA, and this leads to hyperlipidemia. So it's like the synthesis of fatty acids. And also leads to decreased fatty acid catabolism, so the breakdown of fatty acids, and this leads to what's known as the fatty liver disease, and it's called hepatosteatosis. I hope I pronounced that correctly. And next there's some stuff about standard drink. So, as you probably know by now, 10 grams of alcohol or 12.5 milliliters of pure alcohol is a standard drink, and the safe and recommended drinking level is that is less than 4 standard drinks in one sitting. It's considered to reduce the risk of alcohol-related injury, and less than 2 standard drinks per day reduces the lifetime risk from alcohol-related disease. And lastly, patients that drink too much alcohol are treated in 2 ways. So first, remember how it leads to lactic acidosis. So basically your blood pH is too low, so you kind of counteract that with bicarbonate. So you put bicarbonate in and it'll mix with the hydrogen to neutralize the level. And the next is thiamine. So thiamine is used by pyruvate dehydrogenase when it in the link reaction when we convert pyruvate to acetyl-CoA. So this way we try and push it through the pathway of cellular respiration instead of pyruvate being used in lactic used in fermentation to become lactic acid. Yeah. All right, the last thing I'm gonna go through is the great metabolic rates. So this was an active learning. I'm not sure how you guys are gonna do it, probably another Zoom session, but yeah. So it's talking about the energy sources that a runner would use during like a run, basically. So this is a pretty important diagram, and some important points to note is that both carbs and fat contribute the same amount at 3 minutes and 30 minutes. So that's here and that's here. Just something to remember. And skeletal muscle uses 20% of the body's oxygen. It can pretty much metabolize anything to use for energy during the run. And this table is perhaps the most important. Oops, this table. So you want to know that at rest, your body mainly uses fatty acids. When you suddenly need a short burst of activity, such as when you're sprinting, it uses creatine phosphate. When it's moderate activity, you use a mixture of glucose, fatty acids, and ketones. And when it's high activity, you use glycogen, free glucose, fatty acids, and lactate. And lastly, when you're starving, your body will use protein. So this table kind of just goes through the different timing of the race. So at rest, when you're or before the race, the primary energy source of your body is fatty acids, and it's used because they have long-lasting stores and are energy dense. So it's just kind of like how energy is mostly stored in your body. From 0 to 2 minutes, you use carbohydrates and mostly the stored glycogen instead of free glucose, and it's done through anaerobic respiration. Because anaerobic respiration occurs faster and it could be used for quick energy. So glycogen is used instead of the free blood glucose because your other cells still need a bunch of glucose to function. So your body's not ready yet, and it's just taking the glycogen stores instead of taking glucose from all the other parts. And this glycogen is mobilized from liver and skeletal muscle. So we've been through this before. The liver is mobilized and exported through the bloodstream, and the muscle is not exported. In 2 to 5 minutes, we're using carbohydrates still, but now we're using aerobic respiration of stored glycogen and blood glucose. From 5 to 30 minutes, you'll see in this diagram, the use of carbohydrates starts to go down while the use of fat starts to go up. So yeah, the percentage supplies of glucose decreases while the percentage of supply of fatty acids increases. So at 30 minutes, it's when 50% of each is used. So this is the point of interaction of the two sources. And from now on, fatty acids will become the primary source. And it's used through the beta oxidation to create acetyl-CoA that then enters the Krebs cycle. And at 45 minutes, you use stored fatty acids. And they're used due to the lack of oxygen for aerobic respiration and the depletion of glycogen stores. And that is about it from me. Thank you.